in this chapter in a nutshell. So what we started with was the question that how did life originate, right? Where did it come from? Now, at many instances, at multiple places in books, you read these sentences like life appeared on earth at this time. So it gives you kind of a information which is not completely true, that it looks like it just appeared out of blue. So we talked about many theories, philosophical and both observational theories that people used to believe in, and then how the theory of biogenesis, which tells about that bio means living or life, and genesis means it's synthesis. It occurred from non-living molecules that aggregated, and just by sheer luck and chance, these molecules form the building blocks. We do not yet know the exact mechanism how it happened, because uh, this took millions of years back in the prehistoric earth, but we could make organic molecules using non-living molecules or non-living components which were present in the early earth, right? So this kind of gave us a hint that a life evolved, and now we have evidences from multiple fields weaved together. For example, we have seen that similar kind of carbon based long molecules or organic molecules have been found in meteorites and also in asteroid samples, which also tells that the same kind of reaction might be happening, and not might be, like it is happening at different pockets in the universe. Wherever you have these elements, wherever you have conditions that favor, there will be these reactions happening. Now, the next big question is to find life outside Earth, which we have not in the present, like we have not found evidence of current present life, but we have found evidences of that back in past, there are habitable zones which might be having hosting life. When I say life, it's not necessarily the complicated evolved life like we have on Earth, but microscopic life could have been possible on some places. Mars is one of those where we have evidence of water. Like now there is no water there, but we have evidences that back in past, back in time, Mars was a planet with liquid on its surface. So coming from there, we talked about once the simplest form of living system, which is like the precursor of each and every cell. Now, when once that was formed, which we call as the LUCA in science, LUCA, last universal common ancestor. Once that was formed, then how it evolved to give rise to all this diverse kind of life on the planet, right? We also talked about different theories of evolution. We talked about how Darwin observed natural selection, branching descent. We talked about Lamarck. We talked about evidences of evolution ranging from embryological evidence, fossil records, to morphology, comparative anatomy and morphology in plants and animals through analogous organs that lead to convergent evolution and homologous organs that leads to divergent evolution. We talked about adaptive radiation, both in Darwin finches and marsupials in Australia. Also kind of discussed, which if you remember, we talked about biochemical evidence of evolution, which we see everywhere around us now. Do you remember what was the biochemical evidence of evolution? Anyone? Iram or Parker? Parker has also joined in. Good evening, Parker. How are you doing? Am I audible to you, Parker? Yeah, great. So I was giving a recap of whatever we have learned in this chapter in a nutshell, very, very brief and short. So my question was, do you remember what can we call as a biochemical evidence of evolution? What is one thing that binds every life on the planet? That is how our biology works, from enzymes to our genetic material. It does not matter regardless of whether the organism is unicellular, multicellular, it's a plant or it's an animal. Every organism's genetic material is DNA or RNA, right? Um,
Yes.
mostly DNA, and for some it's RNA, for some RNA viruses. But DNA is there, and every life follows the central dogma, which I have already taught you. The flow of information from the genetic material to proteins, like from DNA to RNA and from RNA to proteins. All these things are conserved, highly conserved, which again gives a proof that all life is somewhere connected. It's not very different. It's not that very different looking organisms. For example, you know that one thing that you and a banana plant does, anything that humans and banana plant does in common? What do both of us do to live? Come on, Iram. What do you think of? Yes, right. One is cellular respiration, correct. And when I say cellular respiration, you mean breaking down glucose, right? In the presence of oxygen to produce energy, right? That every cell does,
Yes.
whether it's a animal cell or a plant cell. So what it takes to do this process, which we call as glycolysis, right? Remember glycolysis? It's a 9-step process. It's a 9-step process in humans. It's a 9-step process. The same process is there in cockroaches, ants, trees, everywhere, right? So if life was created from very different designs and very differently, this wouldn't have been same. And the enzymes that we use to do these processes are also conserved. That is the reason for humans, when we have to develop um, multiple times, when we have to develop therapeutic molecules, for example, when we have to develop insulin. So earlier, insulin was extracted out of other mammals' pancreas and then injected for the treatment of diabetes, the symptoms of diabetes. But now, through like genetic engineering, which is the next thing I'm going to teach you. So I'm just connecting the dots here to the next thing. Yeah. Yes, Parker has some doubts. Parker, you can ask. I'll give you some time at the end of the class to ask doubts. Okay. Is that fine?
Yes.
Yeah, we'll discuss that. We'll finish a little early today anyways. So I'm trying to connect dots to the next set of things that we will do. We can do genetic engineering, which call as biotechnology, and we use bacteria to express insulin as a protein, as an enzyme. And this insulin is a protein which is raised in bacteria is the same as what is there in humans, and it works. So the fact that we can make something in a bacterial cell, purify it, put it in the human body, and it works is also a proof that at the biochemical level, life is all related, right? Make sense to both of you?
Yes, sir.
Yeah. Now, in today's class, what we'll be doing is to summarize things and in a broader way and talk about a brief account or brief history of evolution. We'll talk about plants and animals in general, and then we'll talk about humans in detail. And if time permits, a very interesting study which earlier used to be a part of the curriculum for a medical entrance is the evolution of horse. So horses have been, a very good friend and a domesticated animal for humans. But we know that horses are also found in the wild. We call them stallions, wild horse, and most of them are domesticated by humans for their use. So evolution of horse is very well documented, and that also is an interesting thing to look at. But let's start with a brief account of plants first. So this is a figure from your NCRT only. Please, and also for today's class, keep your NCRT open in front of you. Both of you have NCRT? I'll give you time if you don't have to fetch one. You have it with you?
Yes, sir.
Yes, sir.
Okay, perfect. So please come to this diagram. It's towards the end of the chapter, you will find this diagram. And yeah, I think you can see the full diagram now. Now, what I want both of you to do, today's class will mostly be whatever I have taught you, you have to use those concepts and you people will speak more than I will. That's the plan. Let's see if it works. So first thing is this flow chart in front of you, this diagram. This represents, it's a sketch of evolution of different plant forms through the geological period. So whenever we read anything, any diagram, we first have to explain the X and the Y axis. So you see here, what's on the Y axis? What do you see on the Y axis? This is the Y axis. And what do you think is on the Y axis?
Time period.
Yeah, so these are geological periods, right? We call them geological periods. So this is Paleozoic at the very end, the first one. And this is very recent. So this is where you have the present. Right? This is recent, and when you go here, this is the oldest. So Paleozoic, Mesozoic. It's not like geological periods start from Paleozoic. Paleozoic is from where we have evidences of plant fossils, right? Even before Paleozoic, there are like how it might have evolved. There are theories which cannot be tested in lab, but can only be, we can depend on evidences from fossil records. So Paleozoic, Mesozoic, and the Cenozoic, these three periods, then they are subdivided into eras, the Silurian, Devonian, Carboniferous, Permian, and then Mesozoic is Triassic, Jurassic, and Cretaceous. Triassic, Jurassic, and Cretaceous is also the period parallelly. This period was we call it as reign of reptiles, and especially what we call those reptiles who reign the world, dinosaurs.
Dinosaurs.
Right? Yes. So in your last exam, I realized while I was checking those who have submitted their exam, there was a question that when was the peak period of dinosaurs on the planet? There were two options which were conflicting and confusing. Basically, the option Jurassic or Triassic was with the option Mesozoic. So Jurassic and Triassic are subdivisions of the period Mesozoic. They are the eras within that broader category. So if you look carefully, we know that dinosaurs were at the prime here. Now look at the type of plants that were there when dinosaurs were around. Now what I'm going to do is, and we will do it for every concept in today's class, I'll give both Iram and Parker 3 to 4 minutes to look at this. If you want to take the help, it's an open book thing. If you want to take the help of your book, you can, but I will advise you not to go to internet because it will only confuse you more, not help you, because I'm just relying on your honesty and conscience. If you will do, of course I will not be able to do anything, but I'm allowing you the NCRT. I'm allowing you this screen that's projected in front of you. All you have to do is observe this. I will tell you what is on the y and x, and what is on the x-axis. We talked about y, but what is on the x-axis?
Different category of plants.
Yeah, plant forms, right? Different kind of plant forms. Okay, so just observe this. Your time starts now. You have 4 minutes, and you have to write 5 points, the 5 most valuable points that you think, and keep each point one sentence. It's not like you just have five, so you will make 1 point complex paragraph of 4 or 5 sentences. One sentence each. That sentence can be long, but don't make it very long. I want to see what is the 5 most striking things that you can get out of this whole thing. Okay, you have 4 minutes to do that. Is that okay, Parker and let's see what comes out of it. Give me a thumbs up if you're started. Your time starts now. Now I'll be silent for the next 4 minutes and only speak if you have a question. Yeah, cool. So Iram has agreed. Parker, you understood the rules of the game? Perfect. So your time starts now. Begin. Yes, go ahead. Sorry, I was mute. Go ahead.
Sir, what is MYA?
Million years ago. It's acronym for time scale, millions years ago. Okay. If it is BYA, it is billion years ago. Two more minutes. Okay, so got it, Iram, Parker? Ready with your 5 points? Are you there, Iram? Anyone? Ready or you need more time? I can give you a couple of more minutes if you want. Come on, someone say something. Am I audible, people? I think I'm audible. Yeah,
Yes, sir.
right. Yeah, so done. Yes, no, not an exam. It's just we are learning in the class. So no pressures, no worries. So who wants to go first? Iram, let's start with you. If you're okay. Are you there, Iram?
Yes.
Yeah, okay. So let's start with Iram's points. With green, what I think Iram has to say. I'll write it as a bullet point. So whatever your 5 points are. Yes, tell me. What are the 5 most important things you got from this graph, or this representation? Yeah, Iram, you are mute if you're speaking. Are you there, Iram? I think Iram is having some issues or something. Parker, do you like to go first? Sorry, come again. I couldn't hear you.
So I wasn't able to make one.
You weren't able to make points. Come on,
Yes, sir.
you were. I'm sure you made some points. How can you not make any point by seeing of the plant forms to come up were bryophytes? See, it's very simple. You can see that it's coming the first. No?
So,
Yes, Iram. You there?
yes, sir.
Yeah, go
So,
ahead.
Zostero that was the first phylum. It's the oldest phylum that is lying under Paleozoic period. And in this, we have 4 different categories of plant. Xylophyton, Rhinia type plants, Trichophytes, and chlorophyte.
But where is this information on the chart? I don't think it is in the chart. I'm not sure if it is in the NCRT. Is it in the NCRT? What do you
There's
say?
nothing written about plant evolution. It's only about
Right. So that's
they have
why
just
often
presented to me.
schools just skip it. But the idea here is why I do this exercise. What's given to you, what's not given to you directly. If you can infer that, that's also you succeeded in understanding that concept. So you are right to begin with. You talked about Zosterophyllum, saying that it was the first of categories of plants to come up, right? But what about bryophytes, Iram? Do you see here bryophytes? And you see that
Yes,
bryophytes have come up or evolved even before Zosterophyllum, right? They directly connect here to the chlorophyte ancestors, which were the earliest plant-like organisms. If you'll see chlorophytes, you will not be able to say that they are plants. Just like when we see many algae today, we can't tell that, oh, just by looking at them that they are plants. Only because they have chlorophyll, we say that, oh, they do photosynthesis, so let's put them in plant kingdom. So chlorophyte ancestors were the first of the organisms that could do something very similar to what the light reaction in the photosynthesis happens in today's plants. They were not exactly doing the same thing. And why? There is a reason behind it. We'll come to that reason. But yes, about your point, Iram. So Zosterophyllum comes from the tracheophyte ancestors, right? Why we call it tracheophyte? Because now by this time, plants were kind of organized into multicellular organisms, and they have the system which could absorb nutrients from outside by a group of cells or some structure. So that's called tracheophyte ancestors. And after this point came proper plants like Rhinia type and Xylophytans. Xylophyton is what gave rise to majority of plant, even what we have today on the planet. So Xylophytans gave rise to everything that is here, right? From horsetails to ferns, ginkgos, conifers, nettles, cycads, and angiosperms. Everything came from Xylophyton, but something that's still today present on the planet. We know that bryophytes are still there on the planet, right? We can find bryophytes, right, on the planet. And they were one of the earliest evolving plants. That's why bryophytes are so different in everything from angiosperms. Okay. This you can clearly make out if you pay attention to this sketch, isn't it, Iram Parker? Yeah, cool.
so
And any other thing? Yes, Iram, go ahead.
so bryophytes come under the period Cenozoic, right? And that is the latest period, no? So how do you say that this has evolved? This
No,
is the
but
first one.
this is the first one, like the earliest kind of feature. So when I say first one, when we say prokaryotes were the first kind of organisms to evolve, they're still evolving. It's not like bryophytes. All these plants reach till here, right? All these that reach till here are still evolving, right? You're right. Cenozoic is not what we are living in at the moment. It's slightly different from, it's a subtype of quaternary, but yes, these plants are still there. But if you see some other plants that, for example, like this branch that went till here and then didn't rise after that, maybe they just got extinct here, right? Even plants also undergo extinction. And if we talk about grasses, remember grasses? All the grasses that we have, the family, the grass family, grasses are what? In which category do grasses fit? Are they ferns, bryophytes, conifers, angiosperms? What are grasses? Grasses are angiosperms, right?
Yes.
Yeah. So angiosperms till here, like I think till this line, which is between Jurassic and Cretaceous, which is where dinosaurs were ruling, right? In this period, you will see that there's a very tiny pipe. So another thing that you could figure out from this sketch is what do these thickness of the branch. So this is this thick, conifers is this thick. In the same period, you had phenopsids, which were very thin. And in the same period, if you look at angiosperms, they are almost tells you about the abundance, the relative abundance, how much of these were present on the planet. So when I say bryophytes were the earlier ones to come up, it's like they directly after chlorophyte ancestors, they have all the features that chlorophytes might be having, but they do not have features that other plants have, which means uh, if they are 5 - 3, so they never, they never saw the extra 2 in the 5. So what tracheophyte ancestors, Rhinia type plants, and xylophytans have, bryophytes do not have that. For example, bryophytes cannot produce seeds, whether naked or covered. So they are very different from gymnosperms or angiosperms. They have no ability, because that is one characteristic of the gymnosperms and the angiosperms only. Make sense?
Yes.
Yeah. So that's what I mean when I say that they evolved the earliest, right? They came up the earliest. And one thing that you will notice is that angiosperms were almost like very less on the planet throughout till the Jurassic. Something happened during the Cretaceous period that there was a boom. Like now, if you see, the widest branch of plant is angiosperm, and on the planet around, you know, from all the species of plants, majority of the plants are angiosperms. If you count each and every species, you will see that most of the species are angiosperms, right? And though conifers at one point of time were abundant on the planet, now they have reduced because they are now they can only grow in some limited pockets. Same thing happened with, let's say, this thing, phenopsids. This was very abundant in the Carboniferous period and then now reduced in abundance. And for most of the things, for example, ferns, bryophytes, lycopods, ginkgos, it's more or less stable. Whatever they, inflated to, their abundance is stable even till now. But angiosperms, you will see that they just out competed everything, and clearly they ruled the plant kingdom. Yes, Parker has a good question. Why are they getting reduced or increasing? That's the brilliant question, Parker. So this, when we study this kind of evidence through fossils, and we realize that what kind of plant imprints or plant forms evolved and then reduced, this directly tells us about the fact that Earth's climate and Earth's atmosphere was ever changing. It was dynamic throughout. It was not how we get it today. So the atmosphere of the Earth, where there is 21% oxygen, 78% nitrogen, 0.3% CO2, etc., we think because from a few millions of years, like from 3 to 4 millions of years, it has been the same. So we think that it was always the same. It was not always the same. The reason why angiosperms were not in abundance, and the reason why during Carboniferous period, the word Carboniferous means that in this era, carbon-based life flourished. So you will see that either towards the end or in the middle or somewhere in Carboniferous, everything increased. Carbon-based life got a boom, shoot up. We have evidences to suggest why it. But and carbon-based life is not just plants, it is also animals. So the same thing happened to animals as well. We'll talk about it in some time. So They are increasing and decreasing because of conditions that changed. When life started, when cellular life form appeared, in the beginning, the atmosphere was reducing. Remember, that's what the whole point of Urey Miller's experiment was. Yes or no?
Yes.
And reducing atmosphere means that there is no oxygen, gaseous oxygen in the atmosphere. And plants rely on gaseous oxygen, right? Why? Why do they rely on gaseous oxygen? Most of the angiosperms and most of the animals, why do they take oxygen? To do cellular respiration, right? And if there was no oxygen, then how were plants respiring back in the time? And how were animals respiring? So animals were not present when plants started evolving. So plants were the first kind of organisms that evolved, followed by animals. Because around 2 billion years ago, when life evolved, these chlorophyte ancestors, when they evolved, some of these organisms had the ability to release oxygen. Okay. So what they were doing is they were taking water, splitting it, using the hydrogen for themselves. And now you can understand why were they using hydrogen. They were using hydrogen to fix or reduce something in their biochemistry, in their cell, and they were releasing oxygen as a byproduct. Oxygen was not useful to anything at that time. In fact, on the opposite, oxygen is a toxic substance. Today, because life has adapted to live in 21% oxygen on the planet, all the organisms that live utilize oxygen for doing what? What do we do using oxygen? Do we make something by using oxygen? We don't make glucose by using oxygen. If you say that we make glucose using oxygen, you are
breaking down of glucose.
in the process of photosynthesis where glucose is made. What is the source of oxygen in the glucose? The source of oxygen for the glucose is CO2, carbon dioxide. We don't need oxygen because it's already there in carbon dioxide. When plants take water, they split hydrogen and oxygen apart. They remove oxygen. They don't need oxygen to make glucose. You don't need oxygen to make glucose. In fact, you need hydrogen to reduce CO2 to make glucose, because CO2 is CO2, one carbon, two oxygen, but glucose is C6H12O6. You also need a lot of hydrogen to reduce CO2 to make glucose, to fix it. That's why it's called fixing, carbon fixation. Plants fix carbon. That's how they protect all of us from pollution. Lots of CO2 in the atmosphere, plant more trees, they will fix it for you. They will take CO2, use hydrogen, fix it into starch. Everything around us is connected. Do you understand, Iram and Parker? So that's a wrong assumption that we need oxygen to make glucose. What do we need oxygen for? Why do we all breathe oxygen in? Come on. This is what real thing you are studying. I'm sure you all will pass. No worries. But do you understand what you are studying? You know, in many cultures, not in many cultures, almost in every culture, the one who remembers the most is regarded as very intelligent, or is respected. So whether you remember anything, you remember textbooks, you remember tables, you remember facts, you remember religious texts, you remember anything, and you are intelligent. I totally refute that notion. If you remember, you are a hard drive, you are a human equivalent of a pen drive. No glory in that. To be a human is to understand, process, not store necessarily, right? So anything that you remember, if you're remembering without understanding it, that's a failed effort. So why do you breathe in oxygen? Because you remember that you breathe in oxygen, because that's a line in your textbook. You have written it multiple times since your class 4th to get marks, and you must have gotten marks every time. I'm just asking why you breathe in oxygen. Come on, don't be silent. I know that both of you know. I don't know why you don't speak. So yeah, we can go ahead without you both speaking. I can just finish this class. Yeah, anyone? Why do you breathe in oxygen? Or the process of respiration? Yes, what is the process of respiration? That's again another line from the textbook.
So
What do you do? Yeah,
oxygen gives our cell the ability to break down food in order to produce energy, survive.
What do you mean by break down food?
Break down.
I think we did a very elaborative discussion on that, right? We literally use oxygen to do oxidative breakdown of glucose. So we don't need oxygen to make glucose, but in fact, exactly opposite of what Parker was saying, we need oxygen to break down glucose. Yes or no?
Yes, sir.
So oxygen by nature is a very reactive molecule. It is not a good molecule. Wherever it goes, it doesn't help making things, but it helps breaking things down by oxidative breakdown. So you oxidize anything and it will break down. Oxidization, right? That's a chemical reaction. Oxygen, when any organism ages, it produces a lot of reactive oxygen species called ROS. ROS accumulates in our body and we become old. ROS accumulates in our body and we become, weak, ill, immune reactions happen. Lots of, damage happens in the body because of oxygen. In fact, if anyone tries to inhale pure oxygen, their lungs will be damaged. Oxygen is a reactive molecule. It's not very good. In only at 21% where it is what it is now in the atmosphere, it helps you because we have adapted. But there is a real thing called oxygen toxicity. There are bacteria that are called anaerobic bacteria. We will study in detail about it in the biotechnology chapter. Anaerobic bacteria cannot tolerate oxygen's presence in their environment. If you give them oxygen, the bacteria dies because the oxygen goes inside the cell, breaks down a lot of things which bacteria cannot control. And therefore, for anaerobic bacteria, oxygen is a toxic substance. Even for aerobic organisms, oxygen can become toxic if you increase its concentration. Okay. So at one point of time, there was no oxygen in the atmosphere. All the organisms that were evolving, doing very well without oxygen, then some of these organisms started releasing oxygen because they were doing something similar to what photosynthetic plants do now. Okay, they were taking water, splitting it using the solar energy, and then taking the hydrogen, using it for fixing things and releasing oxygen out. When that started, all of a sudden, lots and lots of oxygen started coming in the atmosphere and in the water, which was not good for organisms, and organisms either were dying or they were adapting to tolerate this oxygen stress. And it is also hypothesized with some evidences, but not too much to completely accept this theory. But there are evidences that suggest that organisms, based on what different concentrations of oxygen was there in the atmosphere in the past, there were different kinds of organisms that evolved. One of the questions that everyone asks in evolutionary biology is why in the past, during the Mesozoic era, after the Carboniferous bloom, why there were so big, huge organisms on the planet. And when they all got extinct, nothing like that ever came up again. So reptiles were huge, dinosaurs, they were big, right? The biggest organism on land today is the giant African elephant. And the biggest organism overall, animal, is the blue whale in the oceans. And in terms of plants, you will find maybe the redwood, sequoia tree, or something like that, which, yeah, you know.
Sorry to interrupt. So you said that oxygen, too much of oxygen is poisonous, right? So
Yes,
like during oxygen therapy in asthmatic patients, don't we provide pure oxygen to the person?
we never provide pure oxygen to anyone. So the oxygen cylinders that we get, right? So we all have that notion that if it is an oxygen cylinder, it will only have oxygen, right? So we just fill in oxygen in there and you connect it to someone's lungs and that person is just taking oxygen. That's not the case. Oxygen cylinders, the oxygen that they contain is always mixed with other gases. It could be inert gas like helium or nitrogen, but nitrogen, veins and arteries. So you can use helium, something, but you have to make the partial pressure of oxygen similar to what it is in the atmosphere. Okay. You can keep it slightly up or down based on how, if someone's lungs or someone's body is incapable of absorbing oxygen in the blood, then we might increase that to some extent, but it's never 100% pure. Otherwise, it will just cause oxidation in each and every cell of the lung. Make sense,
Yes.
Yeah. So the therapy that you're saying is to improve the oxygen in the blood. Even for COVID patients, it was done. And for patients who have any kind of lung infections or problem in lung, in case of pneumonia and things, the ventilation that is given to them, we say that they are given oxygen. It's always mixed with some other gas to some extent, right? What it won't have is CO2 for sure, because we know from class 11th now that CO2 and O2 both can compete for binding to hemoglobin, right? Do you understand? In the atmosphere,
Yes.
we have CO2. So patients are struggling by normally breathing. Why? So have you ever thought what is the need that we have to put patients on ventilator? Is it that there is no oxygen in the air? Because the doctor is alive, the nurse is alive, the relatives of the patient is alive, they are breathing in the same air. How is it happening that for the patient there is no oxygen from the atmosphere? It's not that. The point is, in the atmosphere we also have CO2. The lungs are compromised. They are not, taking in oxygen through partial pressure of oxygen. So we have to increase that partial pressure, of course, but we cannot make it 100% pure. That's also toxic. And what we can do is we can provide it with a air which does not have any CO2. So when it will not have any CO2, of course, the partial pressure of oxygen will be higher and it will be the only binding candidate to hemoglobin. So that will increase the blood oxygen absorption. You understand?
Yes.
So that's why we give them oxygen through ventilation. Yeah, cool. So that's a good question. Parker says heart stops pumping for some reason. What is this related to, Parker? Is it related to the, yeah, go ahead, to the incubator thing, ventilator thing, right? See, ventilator is not for heart, it's mostly for lungs. But heart also needs oxygen to constantly work, and brain. So the patient, the brain shouldn't die and the heart shouldn't die because of lack of oxygen. That's why. But that's why the patient is put on ventilation. But ventilators do not Okay, so ventilation doesn't help with beating of the heart. It can lead to dying of the heart for sure. So you are right in that accord that it's important to keep the heart alive. We have to keep giving oxygen. Yes, perfect, great. So yes, Parker, you have any question?
No, sir.
No. Okay, cool. So slowly, when these organisms became multicellular, now, and by this time there is oxygen in the atmosphere, so you need uh, different kind of adaptation to uh, tackle this oxygen stress. And if you know about the theory of symbiosis, anyone knows the theory of symbiosis? This theory suggests like when it was given, and now we know that this happened, this is true by advanced genetic research, is that there was a big cell that kind of engulfed a smaller cell, and later on that smaller cell started living in that big cell. And in today's animals and plants, we call these eukaryotic cells, and they have something called mitochondria in their organ, in their cell. So mitochondria at one point of time in the early ancestors of living forms was a parasite that just entered inside the cell because the bigger cell engulfed it. And what the smaller cell could do was it could tolerate oxygen and use it to produce energy. And what the bigger cell could not do, so it was vulnerable to oxygen. So for this, it was oxygen toxicity. So when it engulfed the smaller cell, they started living in a symbiotic relationship where whatever oxygen used to enter inside the cell, the smaller one will take care of it. And the bigger one, so it will produce energy and give it to the bigger one. The bigger one using that energy will protect itself, find suitable environment to live in. So the smaller one is getting a host, a home, and protection and nutrition. And the bigger one is getting rid of oxygen toxicity. And that's exactly what our mitochondria do in each and every cell of our body. If you know where does all the oxygen go, so in the cytoplasm, we only do glycolysis, right? The electron transport chain, remember that? ETC in animal cells that happens in the mitochondria, right? The inner mitochondrial membrane is where the ETC happens, where oxygen is. You know about ETC, people? Yes, no, because I don't hear anything. You have studied all of this in class 11th, remember? Electron transport chain.
Yes, sir.
What do you used to call that part of respiration? Aerobic respiration, remember?
Yes.
So through glycolysis, which is not aerobic respiration, glycolysis is common in every organism, whether it's a anaerobic bacteria or prokaryotic bacteria or eukaryotic aerobic organisms. Glycolysis is common, but but only through glycolysis you only make 2 ATPs out of one glucose molecule, right? Which is a very less turnover. But through aerobic respirations, how many total ATPs you can harvest? Where you do aerobic respiration in the inner mitochondrial membrane, the electron transport chain, remember that? 36 to 38, right? That's the number. So from 2 to 38 is a huge jump. That's why when organisms started utilizing oxygen, they got lots and lots of energy. And to become multicellular, you need energy. And that is where multicellular life form could, begin. So there was one question which was asked to me by one of my students like a couple of years ago in a batch. Why all bacteria are unicellular? Why couldn't any bacteria become multicellular? And it's a very, very beautiful question, right? Why bacteria? So plants are unicellular, plants are multicellular. Fungi are unicellular, fungi are multicellular. Like yeast is unicellular fungi, when they have multicellular fungi. In case of animals also, we have protozoa, protozoans that are like single-celled organisms sometimes. And we have multicellular animals. But in prokaryotes, all of them are unicellular always. We never see a multicellular prokaryotic organism. The reason is the amount of energy that is required to maintain multicellularity, for the division of labor, for multiple cells to work together, to communicate with each other, to live together as a tissue. Simply, you cannot generate that much energy just by glycolysis. You need a different compartment where you can use oxygen and its reactive damaging power to break down molecules and release energy out of them. And that happens in our mitochondria. And at one point of time, the mitochondria was an independent organism in itself. And what is the evidence to say that? Anyone quickly? You know it. What is very interesting fact about mitochondria? Yes, Parker? You might have remembered this fact about mitochondria. What makes mitochondria special cell organelle? I think class 10th it has
It has got nucleus.
it has its own DNA. It doesn't got
DNA,
nucleus. We not yeah. But mitochondria has its own genetic material. Now the question is, if mitochondria was not at one point of time a free living entity, why will a cell organelle within a cell need its own genetic material, right? The cell's genetic material is enough to control, like in a bacteria, there is only one DNA, one chromosome that controls everything that happens inside the cell. That codes for all the protein that the bacteria needs. It codes for everything that the bacteria needs, all transcription factors, sugars, everything, proteins. But in eukaryotic organisms, proof. And but if someone says that, okay, what you're saying is fine, but just because it has its own DNA doesn't mean it's not of the same origin. So we can very easily check that. When you sequence mitochondrial DNA and you try to match it with the DNA of the cell, like if I take my mitochondria, I sequence the DNA that it has, and I take my nucleus and sequence the DNA that it has and try to compare it, they're not similar, right? They're not similar in how the base pairs are arranged and how the genes are there. They are more similar to a prokaryotic bacteria. So that is another proof that tells us that long back, billions of years back in evolution, mitochondria got engulfed by a bigger cell. And that's what theory of symbiosis was when it was given. And now we have proofs for it. Make sense to both of you?
So,
Yes.
so is there any importance or significance of the genetic material in mitochondria?
Yes. So that's a very good question again.
Because
Yeah.
just 1 minute, sir. When it was engulfed by the bigger cell, it could have evolved, and the genetic material could have just perished out because the cell already has one side.
Right. Very good. That's a brilliant $1 million question here. In fact, that is a question that many labs around the world still are working on, but we know the answer to a great extent. So the answer is why there was a reason why the smaller cells DNA didn't perish, because now it is under a bigger cell, and no need. Bigger cell already has the genome. You are correct. But what the bigger cell does not have in its gene is the ability to tolerate oxygen stress. It has no enzyme that can, utilize or channelize oxygen into some pathway where it can be. It's like a bomb. Oxygen is like a ticking bomb. If you have it in your hand, you also have to control it. If you don't control it, it will just blast and create toxicity and start breaking down molecules. That the smaller cell could do. That's why to begin with, it could tolerate oxygen, right? Our mitochondria, the genome of the mitochondria, the DNA of the mitochondria, code for enzymes that only specifically works in the electron transport chain. And those enzymes are not coded by our nucleus. Make sense to you? Another proof of
Yes.
theory of symbiosis. So it has some enzymes which are different
So we're not able to know.
together, right? Yes, they have co-evolved. You will not allow someone to come inside your house and start living without a prize, right? Will you? You will always like to control that entity which is living inside you, even if it is benefiting you. So over the time, the co-evolution has happened in a way where for the maintenance and, for the maintenance of mitochondria, some proteins are required which are coded by the nucleus. So this was just to make sure in evolution that the bond which is becomes an organelle. But now the cell also needs some control over mitochondria, because mitochondria is clearly controlling all the energy formation thing. So it is the powerhouse of the cell. But what if one day the powerhouse decides that I don't want to be a powerhouse for you any longer, I want independence, right? This can happen because if something can come in and adapt, something can also, try the other. So just to not let that happen, so that life, and because life is intelligent, right? Whether it's a bacteria, it's very intelligent. Bacteria talk to each other. We have now figured out through research that bacteria are not just single entity, wandering around without any information of its surrounding. Bacteria talk to other bacterias in a proper, coherent manner. Okay, but that's a whole new thing for some other day. But yes, they do co-evolve, like Iram was saying, and the co-evolution gave the cell also some control over mitochondria, and mitochondria full control over energy production. Okay, yeah. Is my voice, could you understand, Parker? Should I repeat something? Parker is saying my voice was cracking in the middle.
So it just stops in middle for some reason.
So should I repeat something? Just tell me. Could you understand what I was saying?
Yes, it was clear.
Yes, sir.
Okay, perfect. So yes, so over time, when single-celled organisms became multicellular, now you know how, because energy was no longer a barrier. There were mitochondria with us, so we became eukaryotic. Then came animals, and the first kind of animals that evolved, like proper animals, were fish. Okay, and these fish were jawless fish. So from plants here, let's go to the animal thing. And there was jawless fish, and these jawless fish evolved around somewhere around 350 million years ago. I think that's all in your textbook. So just read that. And by that time, plants were more sophisticated. So we had seaweeds and few other plants that were on the land and in the water both by around 320 million years ago or 310 million years ago. And the first organisms that invaded land were not animals, they were plants. So life started in the water, okay? Because that was the only place where it could have started, right? And life could not start just on barren land or just on air by magic. Like many creationist theories believe that it was just created as it is. It couldn't. So it evolved in water. And then what came to land for the first time were plants. Okay. And animals followed plants on the land. Yes.
So, I have a question.
Yeah.
Dinosaurs, we say that reptiles have evolved from dinosaurs, right? Like a crocodile and lizards. Am I audible?
Sorry, your voice break. I don't know if it's my internet or yours, but your voice broke in between. Can you repeat?
Yes, sir. So I was saying that reptiles have evolved from dinosaurs, like lizards, crocodiles, we say, right? But,
Right.
but you also say that dinosaurs are extinct. So how can a thing extinct and evolve at the same time? That doesn't make sense to me.
Exactly. It doesn't make any sense. And that's why um, teachers who teach very bad biology, adds it, they don't help the subject at all. They just make people not believe in the subject even more because they do a very bad job of teaching evolution. And believe me, especially in our country and in many European countries as well, not just in Europe, and I think across the world, biology teachers do a very bad job teaching evolution, and genetics is the second thing. So you're right. So when we say that reptiles evolved from dinosaurs, evolution simply means new species, right? You need to make speciation events, and exactly this slide will answer your question. So this is your dinosaur. All the dinosaurs somewhere come in this branch. Now this branch will also flourish like this to make Stegosaurus, Paleosaurus, Allosaurus, T-Rex, etc., etc., etc. So lots of dinosaurs, they all got extinct. But what happened is like that speciation event that made reptiles, let's say this lizard. So lizard is a reptile, snake is a reptile, crocodile is a reptile. Let's see where they are connecting with dinosaurs. So this is lizard. I see hair. So this is a common point from where we can go to a dinosaur and a lizard as well, right? So this is the common point. And this happened way before they got extinct. Does that answer your question, Ira? So
Your
dinosaurs
voice
got
is
extinct during
cracking.
okay, sorry. Is it fine now?
Yeah,
Is my
it's fine.
voice? Yeah. So dinosaurs got extinct here. We call it the KT extinction, right? During this period, dinosaurs got extinct. Am I audible clearly?
Yes, yes.
But look at what was the common branching between lizards and dinosaurs. It is here in the Permian around 200 million years ago.
Sorry.
So much before dinosaurs got extinct. Yes, Parker.
Sorry,
Go
your
ahead,
voice is.
Parker. Oh my God. Why is this happening? Wait. Okay, I have shifted network. Is it better now?
Yes.
Yes.
So
Perfect. So I was saying that the branching from where lizards were formed and from where dinosaurs were formed, their like ancestor was way back in time before they got extinct. Okay, so it's somewhere near Permian. So near the Permian era, we have a node from where dinosaurs evolved separately and lizards evolved separately. Now lizards came from dinosaur ancestors. So it's the same myth like humans evolved from chimpanzees. We say a very loose sentence, humans evolved from chimpanzees. But instead we should have said humans and chimpanzees co-evolved from a common ancestor. Dinosaurs and lizards both belonged to the lizard family, the group of reptiles. They co-evolved means reptile or a lizard. Okay, so sauropsids gave rise to all lizards, whether it is a dinosaur, whether it is a snake, tuatara, crocodiles, even turtles. So all reptiles come from sauropsids, even dinosaurs, and they got extinct here. Does that answer your question, Neeram?
Yes, sir.
Yeah. And during the extinction, why only dinosaurs died and all these branches? So if here extinction happened, these branches continued to grow, right? This could be one question you will be asking, and also for birds and mammals, right? So these have different reasons. We will discuss and argue it some other day, but they are also fairly well like understood, not to 100% extent, but we have some understanding that why they survived and dinosaurs died. So starting from early reptiles, as back as 350 million years ago, the early reptiles diverged into two branches. One was called sauropsids that gave rise to all the reptiles, snakes, turtles, tortoises, crocodiles, and dinosaurs. And the other one was synapsids. Synapsids are known as the early ancestors or earliest ancestors of mammals. Okay, synapsids and sauropsids. Sauropsids formed all the reptiles and birds, including dinosaurs, which are no longer there. And synapsids form just one branch, and that one branch goes through therapsids, which are now again extinct, to the present day mammal. So the ancestors of present day mammals, all mammals, us, cats, dogs, elephants, chimpanzees, tigers, all the mammals, the common ancestor back in time was therapsids. So therapsids are all extinct, right? Because of, and therapsids were very small, as compared to many mammals today. They're all extinct, but still the mammal, different kind of mammals flourished. So does that, and here also, they just have shown a straight line, but here also, just before their extinction, something must have diverged, branching descent. That's where Darwin was a genius. Darwin didn't know about all of this, what we know today. But all the evidence together tell us about branching descent that Darwin hypothesized way back just by observing species and trying to understand their correlation, their similarities and differences. So that's where we call that, why some people are genius and why we regard them very high in science. But again, Darwin also was wrong at multiple instances. So was Einstein and Newton and everyone. And that's the beauty of science. What matters is not that who is saying that, okay? But what matters is whatever that person is saying, what are the evidences to.
Sir, I can't
Yes,
hear
Parker.
you.
Am I, Iram, can you hear me? Or you also can't hear me?
So it stops in middle. I don't know.
I think towards the end, it's getting worse. Is it better now?
Yes, sir.
Okay, so let's do one thing. Let's stop here, okay, for today. Go back and finish this chapter from your NCERT. In the next class, I will start biotechnology first, because I think today we just have 5 minutes left and I don't want to start that. But before starting biotechnology, I will take 20 minutes to talk about human evolution first, connecting it with how understanding our genome has helped us to do much more through biotechnology. And that's how we will proceed. Will that work for both of you? Is it fine?
Yes.
Sir.
Okay, perfect. So I'll see you in the next class on Wednesday, and we'll do but we'll start after human evolution, we'll start biotechnology. So bring your questions and your doubts in the next class and finish this from your NCRT. Yes, Parker,
sir.
you have a question?
Yes, sir. I have a question from a different chapter.
Yeah. So you can stay, Iram, if you want. You can leave if you want to stay and listen to Parker's question. That's also fine. Yes, Parker, go ahead.
Sir, DNA and RNA can be double stranded or single stranded, but mostly DNA is double stranded and RNA is mostly single stranded. So I wanted to know the conditions where DNA can be single stranded and RNA can be double stranded.
Right. So see, it is the condition where DNA can be single stranded is exactly when the DNA is being replicated or transcribed. So we know normally, like if you're talking about stable single stranded DNA in organisms, it does not stay as a single stranded nucleotide. So DNA always form bond because of complementarity between the bases, and it stays in a double stranded form. So it is very uncommon, or it is rarely um, unlikely for a DNA to be single stranded, but it is more common for a RNA to be double stranded, right? Now, the as such. So, okay, is my voice better now?
Yes, sir. It just like stops in the middle.
It stops in the middle, right? Just give me 1 second. I'll try this network as well.
No, sir, I don't think it is the network problem. I think it is the mic problem.
Oh, okay. Just 1 second. Let me use the other one. Is it any better now?
Yes, sir.
Okay. So DNA is single stranded whenever the DNA replicates. We know that it opens, right? Both the strands open,
Yes,
correct?
sir.
But for the complete DNA to be single stranded, it it is not, generally does not happen. Okay. Only in pockets where the DNA has to replicate or transcribe. Okay.
Yes,
Artificially,
sir.
you can make molecules like that, but it's very difficult to keep them stable. For RNA to be double stranded, we know of the method called RNAi, RNA silencing, right?
Yes, sir.
So normally RNAs are not double stranded because they have a very reactive 3' oxygen, right, in their structure. But if you make a complementary new RNA, then both of them will form pair and they will become double stranded. So double stranded RNA happens in defense mechanisms where, like in agrobacterium tumefaciens, you have studied that, right? RNA silencing. Some
Yes.
organisms can silence RNA by making its complementary copy and making both of them bind together. So if RNA is double stranded, it cannot be read by ribosomes and it cannot be converted into proteins. Okay. So that is where RNA can stay as double stranded. Apart from that, there are also viruses. In viruses, all different combinations are possible. So there are viruses with double stranded RNA. There are viruses with single stranded RNA. There are viruses with DNA as well. Okay, does that answer your question?
Yes, sir. Sir, can
Okay,
I
yeah,
also ask you to explain something?
yes,
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 sir.
sure. Yeah, no worries. Go ahead.
Sir, can you please explain what is a template strand and a coding strand?
Yes, of course. So this is the, let's say this is a double stranded DNA, right?
Yes, sir.
Now, the coding strand, the name is a little misleading. So coding strand does not code for anything. Okay, just remember like that. So the coding strand is called coding strand. Let's say this one is the coding strand. And this one is the template strand. And why do we say that? We decide it on the basis of polarity. Now, when transcription happens, the DNA dependent RNA polymerase, the enzyme that does it, in which direction it works? Tell me.
5' to 3'.
Right. So if let's say this is the new strand forming, okay, for the RNA, sorry. So it will work in 5' to 3' direction, right?
Yes, sir.
So what should be the polarity of the template strand? 3' to 5'.
5'.
So the strand of the DNA that has the polarity 3' to 5' is called the template strand, and that act as a template for forming the new complementary strand on it, right? And the
Yes.
other strand of DNA, which was 5' to 3', basically it is called coding strand, but it does not code for anything. It is only called coding strand because the sequence that it contains will be similar to the sequence of the newly formed strand. Make sense to you?
The 5' to 3'. That stands,
Yes.
right?
Yes. So the coding strand is 5' to 3', because whatever is the sequence from the template strand. Template simply means, like, you must have used PowerPoint, right? PowerPoint or in the Windows, Microsoft, you have used
Yes.
PowerPoints.
Yes,
So
sir.
when you open a PowerPoint, it gives you some templates to begin with, right? Or when you
Yes.
open Word or Doc, they give you some ready-made templates. Similarly, the template strand provides a template for the process of transcription to happen. Okay.
Yes, sir.
So if the sequence on template strand is A, T, G, C, then the sequence on the coding strand will be T, A,
TACG.
C, G. And what will be the sequence of this new strand formed?
TACG, the same as the coding strand.
Exactly. That is why the coding strand is called coding strand, because it has the same code as of the new strand formed, but it does not codes for anything of its own. Okay.
Yes, sir.
Basically, if you see, it is the template strand that is coding for the new, right?
Yes, sir.
Yeah. So, and that this is the on the basis of polarity. Coding strand is 5' to 3'. Template strand is 3' to 5'.
Yes, sir.
Does that answer your question, Parker?
Yes, sir.
Okay. Any more doubts?
Sir, one more doubt was there, but it is from human reproduction.
Yeah, go ahead. No worries.
For the fertilization to occur, it
Mhm.
should be for the fertilization to occur, it should happen on the 14th day, right? But then what if the sperms enter the female body like before the 14th day? Will the fertilization happen that time?
Yeah. Yes. So see, what is fertilization? Whenever you get these kind of questions, just apply logic. A fertilization is a meeting between sperm and ovum, right?
Yes, sir.
Simply, it's a meeting. Now, sperms can enter into the female system at any given time, right? On day one, like from day 1 to day 5, it's bleeding phase. So obviously, Sperms cannot enter at that time. But let's say from day 6 to day 28, anytime they can enter, right?
Yes, sir.
Yeah. But problem is not availability of sperm. Problem is to availability of the ovum. When does ovum enters into the picture?
At the 14th day.
Exactly. So if a sperm, let's say it's the same thing like boss and secretary or principal and student. The problem is not that you want to meet the principal. So your availability, of course, you are in the school 8 hours a day, 6 hours a day for 5 days a week. What will decide that whether you can meet the principal or not is the availability of the principal. Similarly, if sperm enters on day 12, let's say, fertilization can still happen because sperms can stay alive and they can wait for 48 to 72 hours, right? You went to meet the principal was not there. The secretary said that you can wait for 1 hour and he will come. If you are willing to wait for 1 hour, you can meet the principal. So if the sperm enters on day 12 and can survive for 2 days without getting killed by one of the cell of the reproductive system of female, then fertilization can still happen. Do you understand?
Yes, sir.
Because the average lifespan of sperm, the maximum lifespan of a sperm inside a female's body is around 3 days, 72 hours. But it doesn't mean that all the sperms will live that long because they are all actively being hunted also. So as you go closer to day 14, the chances of fertilization is maximum. 12, 13, 14 is the best day, 15th also, because ovum is released on 14th day. Now ovum also will wait for some time, 15, 16, 17, let's say. And then that will also degenerate. If the sperm comes after ovum is gone, then also fertilization cannot happen. So the prime period is, let's say, day 12 to day 16, 17, right?
Yes, sir.
But this is when we take into consideration that ovulation happened at the proper time, day 14. In some females, not in some, in many females, ovulation, the time of ovulation can shift up or down based on their physiology, their stress conditions, their nutrition, etc. That also happens. You might be very well aware of this fact that the menstrual cycle, as we read in the books, is of 28 days. Is it exactly 28 days every month by the calendar?
No.
No, it shifts. Sometimes you will think that, oh, females often encounter this thing that, oh, it's been 4 days delay from my last cycle and I've still not bled. There's no bleeding, there's no spotting. It could also be because you are under a lot of stress of work, you are traveling a lot. So if you're traveling a lot, you are doing a lot of high, enthusiastic activities that can also affect. If you are not well, if you are under some illness, viral flu, etc., that can also affect. So things change. What we study is a standard. So yes, your question was, if it enters on day 12, 13, can fertilization still happen? Yes. If the sperm manages to stay alive, it can still happen. Okay.
Yes, sir.
Make sense?
Yes, sir.
Yeah. Come on. Next question.
Sir, like as you told the delay in the cycle, sir,
Mhm.
as you told about the delay in the cycle,
Yes.
sir, I have heard from many people like it's because of hormonal disbalance, like doctors also say. Sir, how is it hormonal disbalance? I don't understand.
Yes. So remember when I was teaching you reproduction in humans, I told you that this whole cycle is regulated by a series of hormones. So the first hormone has to come from the head, the brain. Okay. So there is a region called hypothalamus that will produce GnRH. Then it will go and act on pituitary. Pituitary will produce gonadotropins. Then those will travel through the blood and will come to the ovaries and they will produce ovarian hormones. Those will go to the uterus and they will do the effect on the menstrual cycle, right? So you see a lot of, it's like a passing the baton kind of thing, right? So
Yes.
anything that goes up and down, which can happen, right?
Yes, sir.
So that's called a hormonal disbalance. It can happen because of if you are on other kind of medications, if you have a problem in the pituitary, if your pituitary is not producing hormones, or it is not secreting hormones in enough quantity. So that also matters. Sometimes your ovaries are not producing or producing more. So it's a very common thing in today's females to have the PCOD syndrome or the PCOS, polycystic ovarian syndrome or polycystic ovarian disorder. So there are cysts formed in the ovary, right?
Yes, sir.
And a lot of women have this. It's a very common issue. Now, when small tumor and growth of cells, if these are hormone producing cells, they will also increase in number, right?
Yes, sir.
And you will see a lot of ovarian hormone, correct? Or the opposite can happen. If because of these cysts, the hormone producing cells are affected, then the ovary will produce less hormones, right? It could
Yes,
also be nutrition. In a malnourished country, in a malnourished female, in a poor country, the nutrition will decide what kind of hormones you will make. For example, growth hormones are affected when you do not get proper nutrition. So babies that do not get proper nutrition have stunted growth, right?
yes, sir.
Yeah. So all these factors can lead to hormonal dysregulation. It could also be just a simple stress. So if a female is, under a lot of stress and about something, she's tensed about something, too much worried about something, that can also affect your menstrual cycle. And it often happens. Females who are preparing for exams and are under stress and pressure of that exam, their menstrual cycles always fluctuate. It's a very common thing. And then whenever that exam is over, when they are again back to their normal life, it again gets smoothened. Yeah, makes sense, Parker?
Yes, sir.
Go ahead, next question.
Sir, I wanted to ask that uh, in the only one sperm can fertilize, right? The zona pellucida like stops the other sperm from fertilizing. So, sir,
Yes.
I wanted to ask, where does the excess sperm, like the 200, 300 million sperm which enters the female body, where does the excess sperm go?
Where do the excess sperm go? Yeah. So most of them just keep wandering in the female system here and there because, they are given signals by the ovum also to where to come. It's like the ovum is giving them some chemical cues where to come. A lot of sperms come and try to attach, like it is also possible that thousands and thousands of sperm are attaching to the ovum, but only one enters. The moment one enters, the membrane of the ovum undergoes a polarity change. Okay. And because of that polarity change, it becomes kind of a impermeable membrane for sperms any longer. So it does not matter where do they go. Even if they keep sticking to the ova, it's not a problem because they anyways will die after 3 days, right? You know why
Yes,
sperms will die after 3 days? Even
that,
if nothing, yeah, tell me.
that's how long they live.
Why? Why do they die? Other cells don't die. In our body, cells can live for months. Why do sperms die in 3 days?
Because it does not have, the gland and which gives it nutrition.
Yeah, sperms rely on fructose for its nutrition, right?
Yeah,
Fructose is not available in the female body in the reproductive system anywhere. They carry the amount of fructose within it themselves and in the semen from the male system. And that will exhaust in 3 days, around 3 days. So they anyways will die. So it does not matter where do they go. Even if they are in the fallopian tube, they are in the uterus, they are attached to the ovum. One enters, fertilization is done, rest does not matter. Even if they are there, they will die anyways. They don't go exactly, they don't go to some locations, you know. Yeah.
Yes, sir. Sir,
Yeah.
sir, identical twins and unidentical twins, like
No,
how?
non-identical twins.
Yeah, non-identical. How are they like?
So identical, yeah, identical twins are formed when there is only one fertilization. So one ovum, one sperm, one fertilization. But after fertilization, when let's say this is the ovum, this is the sperm, fertilization happened, and then there is this zygote, right? Then the
Yes, sir.
zygote will start dividing into two cells, right? From one cell to two cell, and then from two to four, and so on, right? At this two
Yes.
cell stage, these two cells should attach together and stay together. But sometimes, very rarely, what happens is these two cells after division get separated. So one cell, both these two cells will be shorter, half the size. They will get separated and they will start dividing on their own, right? And they will implant at two different locations in the uterus. So basically, it was just one cell. It got divided into two, split up instead of staying together, and then implanted in the uterus at two different locations, and two babies will be formed. But since their genome is identical, their genetics is identical, everything is identical, like the genes, the alleles and everything, they will be identical twins. Their sex will be same, their eye color will be same, their shape of the ear, face, structure, everything will be same. So that is how identical twin is formed. In case of non-identical twin, sometimes females, so you know that in female, every month only one ovary releases ovum, right?
Yes. Yes, sir.
In January, if it is the left ovary, in the February, it will be the right ovary. But sometimes because of some, again, hormonal disturbance, both the ovaries release at the same month. And when millions of sperms are there, then in both the fallopian tubes, there will be one ovum each, and they both will get fertilized separately. And those are non-identical twins. They both will be implanted at the same time, and they both will be born at the same time. But since they are coming from two different ovum fertilized by two different sperms, one can be male, one can be female, both can be male, both can be female, but they will not be identical looking.
Yes, sir.
A non-identical twin is more common than having an identical twin.
Yes, sir.
Okay.
Sir, one more thing. I'm so sorry. Can you please explain the figure 3.8, the female part, the B part?
Yeah, go ahead. Go ahead. Come again. I missed you. Yeah, I'll
Can
go
you
to.
explain
Can I please?
explain the 3.8 figure, the B part, the female part?
Okay, just 1 second. Let me open that figure.
Page 49.
Page 49. Okay. 46,
The B part.
46, 47. 49B part. Yes, great. So basically, you're talking about. Yeah. So the B part is in the blue, right? Which is oogenesis.
Yes.
Yeah. So that is simply what it says is that to begin with, there is oogonia, oogonial cells, right? That divides by mitosis and forms primary oocyte. Okay. So these primary oocytes are again diploid cells, and then they are the first cells that undergo meiosis. So you can see that after primary oocyte, they are showing first meiotic division, right? But the problem, but the thing is, this meiosis does not complete. It only reaches to a point where there is this white cell. You see the white thing before birth line?
Yes, sir.
Yeah, that white thing is the arrested oocyte. So oocyte gets arrested at prophase 1 and before birth and stays arrested till the baby reaches puberty. So after birth and before puberty, the primary oocyte stays arrested in prophase 1 of meiosis 1. And when puberty hits, the first meiosis completes and it forms the first polar body and a oocyte, secondary oocyte. Now this secondary oocyte is what is released from the ovaries, right? And it still has to go one more round of meiosis, because you know that meiosis has